a lot of people think it's like a bigger deal than it is, but to me it's just so much, it's so normal. Like, everyone I know is in a lab that deals with cancer in one form or another, or some sort of other disease related. My name is Anna Whalen, and I'm a senior at the University of Illinois in Urbana-Champaign, and I am studying bioengineering. Bioengineering is where life sciences meet engineering, and especially in my case of bioengineering, I'm on the cell and tissue track, so I look at how cells interact with your body and how they respond. That has a lot to do with like cancer research and stem cells, and it's really like front of the line research in those aspects. In the past, I did spend one summer interning at Northwestern's University, their Robert Lurie Cancer Center in Chicago, and there I kind of learned a lot of, first of all, how to conduct research, getting used to kind of like the culture hoods and benchmark research and kind of how to go about the aseptic technique. But within that, they were looking at leukemia and typically dealing with like a few drugs and seeing how those react with cells. Right now, I'm part of Gregory Underhill's lab, and there I'm studying lung cancer. So tomorrow I am adding drugs to the seeded arrays. What we do is we have the slides and we add hydrogel, and then we incorporate with an arrayer these tiny spots, and they'll have different ECM proteins or combinations of them. And then we take the cells and we seed them on top, and they'll adhere to those certain spots. And then after that, we incorporate the drug media at different concentrations, and we're again dealing with the cisplatin and sunitinib, and we're doing a dose response array curve. So what I'll be doing tomorrow is adding those drugs. So I'll have to calculate the proper concentrations and then create them and then add them. Right now, I'm looking to see if the cells adhere onto the spots. They were seeded about 3 hours ago, and there's formation. There are two lines of cells right now, A549s and HCC, and so the A549s take about 3 hours to adhere on those spots, but the HCC take at least 4. But it's been 3 hours and there's not great spot formation, so like adherence, so I'm gonna see what Karen would like to do.
Wow. Yeah, we have to wait longer.
How much longer should I wait?
Like, those don't look like good arrays to me.
How much longer?
Like 2 hours at least.
2 hours?
Oh, but some of these are good. Like that's ASCL1. Yeah. We do have results. So it's a very difficult analysis to do. What we see is that when you treat cells that are exposed to collagen 1 with a drug, they'll survive. When you add another matrix protein called tenascin C to collagen 1, the two of them together are cause for cells to die in response to drug. So that's interesting because that means the context really matters. Cancer is not one disease. This is a commonly sort of stated. This is a common statement that people make. It's many diseases. I'm not trying to cure cancer. I'm trying to find ways that we can tackle certain aspects of it and make things easier. It's a very incremental process.
Unfortunately, as much as people may think that curing cancer could be plausible within the next 5 or 10 years, it's a pretty complex process, and doing research is making me realize that you can spend months and months on one experiment and just come out with like no results. And not necessarily that it's bad, it's just eliminating another option, per se. I mean, there's so much research out there, and it's crazy to think that with all the research available, there's still so many unanswered questions. So the cancer, problem definitely has a long way to go. And so it'll be interesting to see how those advances come.